All right, we're going to go ahead and get started. I want to welcome everyone here today to the first conversation of vested interests. I'm Lou Basenese, Public Ventures President and Chief Market Strategist. I am joined, thankfully, today. I'm in Boston, even though it's a tough for a Yankees fan to come to Boston, but I did it for these two gentlemen because they're good guys here. So I'm joined today by the CEO of Cue Biopharma, Dan Passeri, as well as the President and Chief Scientific Officer, Anish Suri. So welcome, gentlemen.
Thank you, Will.
Appreciate you joining us today. Before we get to the conversation, I do want to make sure that we cover some housekeeping items. We are going to be making forward-looking statements, or we may make forward-looking statements today. We ask that everyone review the recent filings from Public Ventures and MDB Capital Holdings, our parent company, that filed with the SEC for certain risks and uncertainties. By the same token, I do need to make the same cover statement for the executives here from Cue Biopharma. We just ask that you refer to their latest SEC filings to cover those risks and uncertainties. And then last but not least, we have to, for FINRA regulations, disclose ownership. So I personally own Cue Biopharma. My family owns it. Our firm owns it, and we have done investment banking for them in the last 12 months. So, gentlemen, now I've kept us out of the legal gray areas, and Dan has a legal background. You can probably appreciate that. I want to start today in somewhat of a little bit unorthodox space because everyone's very familiar with the data. And when we talk about investors, we always focus on numbers first. But what I think is really critical here with Cue is the patients, right? This is really about treating and hopefully curing different types of cancers. And the good news is, in Cue's case, we're seeing really strong data in that sense. We're seeing patients live longer. They're living a higher quality of life because of the less side effects or really no toxicities that we've seen to date yet. And we're seeing that expand across indications. So from that human side, I want to ask both of you. Dan, you might have a few more gray hairs than me. You've had a very good career, but you're now here. You've spent the last 7 to 8 years with Cue when it started as an idea on a napkin for all intents and purposes. What brings you here from the human side? Is it personal experiences with cancer? What is your passion that's driving you as an executive here?
Sure, and appreciate the question, Will. It's actually refreshing to have it asked that way. First of all, I'll just start off with the fact that this is a human endeavor. Cancer is a horrific disease. It still remains with us after Nixon proclaimed war on cancer back in the 60s. It is still
I wasn't alive then.
Yeah, all right, well, I was with the gray hair. And, every line we see that we're showing on a graph, that's a human being with a family. And everyone on this call will be impacted somehow by cancer. So from my personal perspective, my entire career has been in oncology, developing cancer drugs in some form. Started off at the National Cancer Institute, literally back in 1990. Worked in the day while I went to law school at night, and had the privilege of working with a lot of the luminary scientists back then. And the reason I joined this company when it was formed was the idea that we could activate the human immune system, the patient's own immune system, to recognize cancer as foreign and have the patient's immune system attack cancer. And that is the therapeutic rather than these cytotoxics. And if we all reflect on it, we're really using arcane technology historically, where we're exposing cancer patients to literally poisonous chemicals that are more poisonous to rapidly dividing cells than your.
Right. We kill the cancer before we kill the patient.
That's exactly right. And that's the reason patients lose their hair. They have horrific GI distress. Their hematologic system gets really messed up. So this is a paradigm shift, and it represents a new frontier in cancer therapy. And, I really feel privileged to be part of this. I don't want to take too much time.
Anish, I want to come to you because you arguably are, well, definitely the smartest guy in this room right here. But no, you're a luminary in the field. You came from big pharma, from 5-6 years at Bristol Myers, 5-6 years in the global capacity at Janssen, where you probably had unlimited budgets, unlimited visibility into compelling things, and then you chose, to come here and work with Cue. Can you give me some motivation, the personal side of that? What made that decision real for you, and why you made the change?
Yeah, I mean, look, Lou, so my background's in immunology, as you're well aware. I was on the faculty at WashU School of Medicine in St. Louis. Just as a very sort of frank disclaimer, I had no intentions of being in industry, none. I was actually looking to move the lab to Singapore once upon a time, and through a series of connectivities got connected with Bristol Myers Squibb, which was the only company at that point doing things I was being interested in. And that's what our interest has maintained for about 30 years, which is really understanding how does the immune system make the decision between self versus non-self. At the highest order, that's the single most important decision the immune system has to make. If you can sense cancer as non-self, you react to it and you destroy it. In autoimmunity, you sense your own tissue as non-self and you react, and you should not react. Right. So that's a fundamental principle. So went to BMS, serendipitously and fortunately was a part of the team that led diligence on Metarex, from where came the first checkpoints, the foundational work on ipilimumab, Nivo, and through a series of sort of wonderful collaborations, really, we were able to bring these drugs to the market as the first approved checkpoints, in large parts to the team there. Moved on to J&J initially with the site in Pennsylvania and in La Jolla, California, and then on a global sort of assignment to Belgium, which is where I was when Dan had reached out. And what drew to me at that point is the company had an entirely different way of looking at the problem. And the problem fundamentally lies in the following situation. And this is the analogy I've often used, is you do not need to light your house on fire to light up your kitchen stove.
Yeah.
And Q had a technology that could light up the kitchen stove. So if you can only selectively perturb and specifically only activate those T cells that are relevant for cancer while sparing the carpet bombing, you automatically dial in two very important things for the patient. You dial in specificity and selectivity, and you dial in safety and tolerability.
And that was enough to convince you to move your family, kids back from Belgium. And I understand live in a hotel for a couple months.
Yeah, Lou, I never thought. So, as these things happen, with the large pharma, they moved our entire house from Pennsylvania, where we lived in Bucks County with 3 kids in the international school. I'd actually extended my assignment by another 5 years. And when I resigned, I had to we had to manage this whole thing back here, but it was worth it. But it came at a lot of personal trials and challenges. I mean, moving back here took about 60 days to ship our home from Antwerp to Boston, living in a hotel for about 64 days precisely.
Yeah, approximately.
Test the limits of your marriage.
But I think this is important, right? This is the human side of it too, with why you're committed to what you're doing. You saw something that was a paradigm shift, in your words, that motivated you to move back from internationally. So I think there's a good dovetail into really the data, right? I mean, this is what we've seen, and I want to share. I don't want to belabor any of the data because you both have done a tremendous job of getting it out there. Most recently at SITC on the conference call, the numbers speak for themselves, but I want to make sure that investors have the context here for what we're seeing on the data side. And so I'm going to briefly share this just to guide this conversation. So right now I'm showing the slide that's showing the median overall survival for Q101 as a monotherapy. And I think, you're seeing here the big pronounced versus Keytruda in second line. I mean, we're more than double, almost, approaching triple the expected survival rate. And then if we look at it in combination with Keytruda and different CPS scores for the benefit, we won't go into that specificity here. Let's just keep it general, is that you're seeing double or triple the rates again. So talk to me about this data. What does it mean? And then more importantly, do we have some updates here?
If you could go back to the previous one, let's start there. I just want to make sure everyone understands this is monotherapy, meaning our drug alone is infused into a patient. And the objective is that it is stimulating the patient's T cells specifically against a protein that's on the cancer. In this case, it happens to be a viral driven cancer. So it's a viral protein. So the drug does nothing to cancer itself. It's stimulating the patient's immune system to recognize cancer as foreign, as Anish stated. And what's really important about this slide is it's showing that Keytruda in second line, this is when they first present with recurrent metastatic cancer, that's front line.
Right.
This is before Keytruda was approved for front line, okay, which is now the standard of care in front line. Second line is when they were resistant to that frontline therapy. So their median overall survival is 8.4 months or 7.5 months with Nivo.
Yeah.
And what you're seeing here is in third line and beyond with our drug. So these patients are even worse. We expect that their median overall survival is probably around 6 months.
Right.
That means 50% of the patients won't live past that date. Okay. And what we're seeing is a median overall survival in third line and beyond of about 21 months, 20.8 months.
And growing, right?
And let's just even say that's where it ends up, but it's still emerging. Yeah. That is a profound development. So again, it's the patient's own immune system that's recognizing cancer as foreign. So Human side, let's remember, these are human beings with families. They're now experiencing a holiday where they would not have. They're also having a much better quality of life with a drug that's not a cytotoxic.
And a significant extension. And I think it's worth noting here too, like Keytruda is a $21 billion a year drug that's used obviously in other indications besides this. Opdivo, I think, is about 10 billion. So again, it points to, if you can have a similar effect, and I'm going to ask you questions about this later, in other indications, it could open up the opportunity for Q101 to have similar impact.
Now,
So
you can
is
go to
there
the next
anything-
slide, and I'm going to ask Anish to elaborate here with the CPS scores. This is in combination in frontline. So, right, we moved from second line where Keytruda was being used. Now it's being used in frontline as standard of care. It received approval because it extended the median overall survival for these patients in frontline from around 10 months to about 12 months.
So only an additional 2 months.
Approximately, yeah.
Right.
And remember, Keytruda liberates the existing T cells to attack. So the patient has to have activated T cell trying to attack the cancer. What we're seeing is a profound enhancement in the overall response rate, meaning we're increasing that arsenal of T cells trying to attack cancer. So Anish, if you don't mind elaborating on the CPS score.
Yeah, I think one of the things, when I look at this data, it becomes very apparent, and I hope everybody appreciates this, on what a dire unmet need there is in head and neck cancer. You look at these survival months, this is not a favorable outcome, right? I mean, when you're looking at a median survival of a second line checkpoint at even 8 months, that is not a lot, just to put it in perspective, when you look at the human element of what's at stake here.
Yeah.
Moving that to 20 months and with a better quality of life, a longer life, is a meaningful. Just what you pointed out, Lou, first line approval is at 12 months, essentially a 2-month increment over, over sort of the standard of care. Now, what happens here is you already see in the response rate in all patients. So in order to receive Keytruda, the tumor has to express the protein for Keytruda, which is PD-ligand. You have to have the target.
Right.
And that's that CPS score, essentially.
Right, it's saying whether or not you have the target.
Yes, you have the target. And if you have the target, you get the drug. And most patients, 85 to 90% of them will have the target. So most patients in head and neck cancer are eligible for checkpoints. However, there's the ones that have the high target, which is CPS high, and the low target. Keytruda has an overall response rate of about 19%.
Right.
But if you look at the low ones, it actually responds even lesser there. It's about 14 to 15%.
And that's Keynote 48.
That's Keynote 48 study.
Thousands of patients that have been on it.
And the reason we point that out is 50% of head and neck cancer patients are CPS low, which means when you walk into a physician's office and you look at statistics, your chances of responding are 1 in 6.
Wow.
Okay.
So a huge unmet need.
Huge unmet need. And in that population, we're above 50%.
Okay.
So, for the treating physician, it becomes an enormously attractive proposition, which is one of the reasons why we've been able to recruit from these excellent medical centers, when you can actually, if your patient has a 50% or greater chance of responding to the combination, as opposed to 14%.
Right.
That's a pretty significant change.
Meaningful for patients. And then let's bridge the gap to, you said, significant unmet need. And I think investors have expressed to us in various forums that they think this indication might not be that big of a market. In your most recent conference call presentation, you added this slide, which I think is instructive here, where it's actually, points to how big these markets could be for Q101. Can you take a moment to comment about, just to respond to the people that say it's a limited market size?
So from the science, there's 2 things that have to be pointed out. One is, among a variety of sort of cancers, the HPV-induced head and neck cancer is actually growing in the Western Hemisphere.
Okay.
It's almost doubled in the last 40 years. Okay. The HPV-negative cancers have actually gone down. That speaks to lifestyle.
And to be clear, Q101 addresses HPV
positive. That's right. So this is
referring to about, I'm sorry, just want to clarify, about 70% of oropharyngeal cancers are HPV positive now.
Okay.
It used to be predominantly EGFR.
Okay.
Right. So that number continues.
So just when you think about the disease, this is not a minor fraction. This is a significant fraction and growing.
Right.
All right.
So that all told here, I think if you add in locally advanced and expanding into other HPV positive cancers, this gets us to a multi-billion-dollar.
This is a multi-billion-dollar opportunity, no question about it.
Yeah.
We're right now only talking about recurrent metastatic late-stage cancers.
Right.
Either monotherapy in extremely late stage or recurrent metastatic frontline in combination with pembro.
Okay.
A lot of patients are diagnosed in the locally advanced stage, and that's where we are moving.
Yeah.
That's the adjuvant setting, the neoadjuvant setting. And that's a massive market opportunity. You take that totally together, it's a multi-billion opportunity. Now you look at other HPV-driven cancers. Cervical is 100% HPV-driven, anal, penile, vulva. So you have now market extension opportunities.
It's a great initial proof of the platform, and then expansion, and it represents a big market.
That's right. From a life cycle management, Lou, you have multiple indications and multiple lines where you can expand.
Okay.
For very attractive economics. And that's been a part of our strategy. Look, you got to generate the data. Data is the only currency in this business.
Understood.
And we have generated data with almost 80 patients dosed here.
So any data updates that are relevant since SITC's release, or any comments that you can make about the data?
Sure. Is the slide still up?
We can bring it back up.
Yeah,
for
bring
you.
it back up. I just want to emphasize something and then I'll get into it. Thank you. So important here is this first study is the HPV-driven cancer, and it's underscoring a number of principles. What I want to emphasize is the monotherapy in third line and beyond. These are salvage patients. They've been exposed to multiple other therapies. They're not in good physical condition. These are the worst of cancer patients, and we've extended life from 8.5 months to 20 months plus as a monotherapy, stimulating their immune system. As Anish just stated, the strategy is then to move upstream into frontline with Keytruda, where we have shown more than a doubling of overall response. In low CPS scores, we've tripled the response rate. Progression-free survival looks like it's at least doubling plus. That's still emerging. Overall survival, it's too early to tell, but that data is looking very encouraging. And what you just asked, the data as we continue to survey these patients is holding and looks like it's becoming even stronger. So that's really important. Now, as you have on this slide, as a strategy, we're then going into the adjuvant setting.
Right.
So what we're doing is going from salvage patients who are in the worst physical condition, and we're showing the drug works and it's extending life in a very demonstrable manner. Moving upstream, we're seeing even better data. And now we're going to be moving upstream even further to the adjuvant setting.
This is the initial diagnosis.
This is the initial diagnosis. Their immune system should be in better condition. The objective there is, in essence, to hopefully go for cure in a significant number of patients where they don't have recurrent metastatic. And what happens is they remove a tumor, that's your primary tumor. They put you on chemo, radiation, that's historical.
Right.
And then you end up with a significant percentage of patients where it reemerges within a 5-year period.
Right.
That's because you have micrometastases. You have cells that have migrated. You can't see them on a scan, and then they emerge. Well, if we can stimulate the patient's immune system to recognize those cells as foreign,
it can eradicate.
and it can eradicate it. So I just wanted to emphasize that's why we talk about a paradigm shift. It's changing the landscape.
So then let's make the jump from Q101 to Q102, because I think what's important and instructive is not just the additional market opportunity that it opens up, but the work and the data set that you've gotten from Q101. You would believe that it would play out the same way with 102, and that's happening, right? So talk to us about how the first indication was the longest and the hardest, and then each successive ones like Q102, why this becomes more compelling as you start talking about strategic partnerships and market opportunities.
Let me just talk about market opportunity, and I'm going to turn it to Anish to talk about the modularity.
Okay.
And how the science basically allows itself to be modified for different indications, et cetera. So HPV as the first sort of foundational data set is to show that we can target specific T cells, delivering an immune activator to only the T cells that matter. And we've demonstrated that with 101.
And we've used the analogy that's like a precision laser guided missile versus carpet bomb.
That's exactly right. And one of the things you see when you are treating patients, particularly in a phase 1, there's a lot of competition. Physicians are using these experimental drugs because they're trying to give their patients hope. But as they start to see the data emerge and that data starts to become enriched and starts to convince them their patients are actually benefiting, it becomes easier to enroll patients, and that's what we've seen with 101 towards the end
So
of
you
that
crossed
study.
that tipping point, that chasm of
Yeah,
getting the buy-in from clinicians.
We were aiming to enroll 20 patients for the phase 1B, and that towards the end, we had a demand of sort of increased pent-up demand for the drug. So we ended up treating 25.
Okay.
And that laid the foundation of the platform's mechanism of action. 102, it's exactly the same molecule, except for an epitope, a 9 amino acid sequence. So 99% of the molecule is the same, except for the targeting moiety, what T cell population it targets. So the market there is much larger. It's called Wilms tumor 1. It's expressed in a significant set of solid tumors and hematologic cancers. It's called an oncofetal protein. It's a protein that's expressed during embryonic development and then typically quiescent or quiet in the adult, except in cancer. You see it basically recapitulated, turned on again.
Okay.
And we're now targeting cancers that overexpress WT1, and we're seeing the drug actually play out as we would have hoped. I'm going to now turn it to Anish to talk about the modularity of the platform in WT1.
Yeah, I guess the question leading into this, have you seen anything that you didn't expect with from 101 to 102? And should that translate into 103, 104 when you specify those targets?
So the best validation for this, Lou, is actually from the FDA, because this is the world's foremost regulatory agency. And when we went to the 102 IND filing to the FDA for the Wilms tumor 1 molecule, we referred to the safety and clinical data from 101.
Right. You didn't have to do any IND enabling
studies, And convinced
right?
the agency for the first time in the history of my career doing biologics, we did not have to do any additional toxicology.
Okay.
And we were allowed to start the 102 trial at a clinically active dose.
So do you think that significance is lost on certain? I mean, you just said the first time in your career.
I think
you didn't start yesterday. So that's
I think as we talk with partners and folks who are in the drug discovery and development business, they recognize this immediately.
Okay.
And that becomes a huge part of de-risking and validation and strength for the platform. Because remember, every time you bring a new drug in, the agency expects you to do a de-risking toxicology and a dose escalation. In the case of 102, as an example, we saved ourselves about a year's worth of dose.
You got to start at the initial dose of 1 milligram per kg, right?
So when you look at development timelines, cost, patient benefit, most importantly, because patients in the lower doses are not benefiting as much as they would from an active dose.
Right.
You've actually hit all those metrics. So the best validation to me is not what we say, is what the agency reflected.
Yeah. So let's bring that back though. So the agency validates it, right? They're the gold standard bearer in that. And now you're saying a comment you made is that the potential partners in the space, the other players in the pharma space are recognizing that as well. So let's talk about that because I think there's a disconnect sometimes between what investors see as the development timeline, how long it's going to take, how much money it's going to cost, and what potential partners see. Again, you worked Bristol Myers, Janssen, and part of J&J. Is it accurate to say that they look at it and see it now as a platform potentiality that can have a lot of opportunities? I think you use an infinite number on the last.
Yeah, I think
are they seeing it that way?
I think the scale is what's being appreciated, but a lot of that appreciation has come from the recent data, the regulatory validation, the clinical data, and the fact that we can generate essentially an infinite number of molecules too, because all we now have to do is change the tumor targeting from HPV, Wilms tumor 1, KRAS, MAGE, PRAME, and the vast majority of the molecule remains a constant. So this de-risking has a read-through to the entire pipeline, which is a big win. And I don't know of another platform that has actually ever demonstrated that. Every time you make an antibody to a different checkpoint, you have to do all these studies all over again.
Yeah, and for the benefit of everyone listening, I know there's a lot of alphabet soup here, but it's basically just different cancers that you can target with the platform.
That's right. So let's take Wilms tumor 1. We went in these first 4 indications for 2 major reasons. One, there's a very high unmet medical need: colorectal, gastric, pancreatic, ovarian. Number 2, these are indications largely which have been refractory to checkpoint inhibitors. They've
Right.
been tried and failed.
So nothing's
The
working.
Keytrudas,
Nothing's
the
working
Nib-
right.
Yeah, so these are multi-billion dollars that failed.
Yeah.
We already in dose escalation are seeing anti-tumor efficacy. So now you turn the tables, the leverage is with us because this is a greenfield
So
opportunity.
it's turning the tables. You talked about a turning point, and Dan's about to turn the tables on us. He's being a Red Sox fan over here. He's Nancy with the Yankees fan over here. But talk to me about the shift, right? Anish said it was as more data, the validation from the FDA. It sounds like there's been a shift in the way partners view you and the data. Is that accurate? And if so, how would you characterize what the change has been?
I'd characterize it, Anish uses this phrase that the data is your currency. Well, it's easy to say something in theory. And we were basically presenting the model of a platform that was modular and scalable. But now we have data showing that going from 101 to 102, it was just a shift in an epitope, a 9 amino acid sequence. So 99% of the molecules the same. The FDA bought that logic. They view 102 as an analog of 101. The data from the clinic from 101 was accepted as a proxy for what they would expect to see with 102. That is a profound sort of foundational shift and acceptance of this premise. Now, what's important is this metaphor of a laser-guided missile versus carpet bombing. Well, in a laser-guided missile, the coordinates determine where you're targeting. So what we're doing is using the same warhead, basically, but we're changing the coordinates with each cancer type. And that's what the 9 amino acid sequence is. It's targeting.
And
different
now,
T-cells.
I guess the question is, did bigger pharmaceutical companies not believe the potentiality because it hadn't been done before? Well, you saw, I mean, CAR-T was all the rage starting in 2017, and now more recently we've seen that 20 cases of secondary cancers because it's broadly targeting or activating versus specific. Has that what's changed? They figured out, hey, the data supports the hypothesis, and now they believe the data, or is it more profound than that?
No, I think it was a couple of things, and Anish, please chime in when I elaborate on this a little bit. Pharma sector was looking historically for one size fits all, a blockbuster drug. And that's why they invested so heavily in IL-2. If they could stimulate cancer,
so the Nectars of the world,
the Nectar, Synthorx,
or whatever. And
the problem is they were indiscriminate activators. It was what Anish refers to as lighting your house on fire to light the stove.
Yeah.
Well, they were injecting a bolus of IL-2 systemically circulating through your body. That's not how nature has designed IL-2 to be used. We found a way to deliver it only to the target T cell that's relevant to cancer. And we're not seeing all these deleterious side effects that you see with indiscriminate IL-2. So there was a shift from pharma wanting that one size fits all, where we were under the radar focusing on very selective, targeted deployment. And now the field is moving in that direction. Because they didn't see any of the therapeutic promise.
So the great Wayne Gretzky quote, go to where the puck's going to be, not where it is, right?
And that's what
you
we're
guys
seeing
were there, and now you're seeing that transition.
So the data we have for 101 and now emerging with 102, we're getting that attention. In the field, we're seeing pharma companies actually starting to use targeted approaches, trying to find ways of targeting specific T cells. So our platform is becoming more and more, let's say, in focus.
Okay.
And you want to elaborate on that?
No, I think you've covered it very well. I mean, the fundamental challenge is selectivity and specificity, and you have to accomplish that. The other fundamental challenge is that in a checkpoint to work effectively, you have to have the right soldiers in place, which are the right kinds of T cells.
Yeah.
And when you're a physician, if unfortunately you're a cancer patient and your physician gives you a checkpoint, they are making an assumption that you have the right T cells and hopefully this checkpoint will work. But we know in head and neck cancer, only 19% of patients have those. The rest don't.
Yeah.
So we become an automatic precursor for the checkpoint to work better. So we've actually already demonstrated that enhancement with Pembro in head and neck. And you look at the numbers of indications where checkpoints are approved.
Yeah, this is what I was going to bring up now. So this, I want to, again, to drive home this point too, of you've established where it works, how it works. And now we look at these yellow dots here for the benefit of everyone. These are FDA approved drugs, these types of indications with, overall response rates with, I'll qualify it and say they're pretty paltry, right? 20%, it looks like the average. So
Most
you're-
of the average is
yeah.
around 20%.
Yeah, right around 20%. And if you use head and neck as a proxy, the potentiality for Q and their platform, it's double, possibly tripling those overall response rates. So talk to me about, let's bridge the gap into strategic discussions, because I think on the last conference call, I'm kind of a nerd and numbers. You mentioned partner or partnering 8 times, which is a 100% increase from the previous call. So that I don't think is a coincidence, because you said there's been a shift in how they view your platform and where they're focusing. What does this look like? Are these all indications that could possibly be monetized in partnering relationships or business development discussions? Because I want investors to understand what the partnering potential is, because I think there's a perception, you mentioned it before, a single type of asset, right? An IL-2. If you partner that away, then you've given away the farm, as opposed to preserving stuff for shareholders, which I think is always a key focus.
So as a backdrop, let me just add, without the shareholders that have invested in the company historically, we would not have been able to get to where we are. So where we are now is this slide is really important. It's showing the landscape, and those yellow circles and red circles, that represents the overall response rate of checkpoint blockade. Which is basically PD1, PDL1 axis, for instance. And you can see on the left, the most immunologically active is melanoma, and then renal. And these are 2 immunologically active tissues. And as you progress to the right, the numbers get even lower and lower and lower. And each one of those cancers have proteins on them that are being presented that are unique to the cancer. And the key is identifying proteins that are on the cancer and not on your normal tissue, right? Whether that's a mutated protein or an aquafetal protein or a viral protein. So what you see there with 101, with head and neck, it was a cancer that had a 19% overall response rate with the frontline standard of care, and that's Keytruda. We were able to increase that to 47%. And again, that data is still emerging, but it's looking even more robust in terms of the numbers that we're seeing. So we basically more than doubled the response rate.
Right.
And what we did is stimulate the patient's immune systems. And that's the sort of foundational example. Now, if you go over to the right with 102, Wilms tumor 1, it's overexpressed in all of those cancers that fall under that bar. And where you see a red circle, those cancers have basically not yielded approval for checkpoints.
And that's why you're having the ability to recruit patients in those indications, because there's really nothing else that's out there.
That's right. And as Anish stated, these represent big markets. And if we're activating or inducing the patient's immune system in these immunologically cold or tepid cancers, that is a big deal. That shifts the landscape where we now have the leverage.
Yeah.
Because now the checkpoints need us to reach those patients.
All right. So let's talk about that change in focus. If I look here, just roughly, it's about 15 different potential indications. Earlier today, let's bring it to like absolute recent news. In the last 24 hours, Gilead strikes a deal with Compugen for anti-IL-18 monoclonal antibody, right? That's about as scientific as I can get. But 60 million up front, 30 million near-term milestone. The stock responds by rallying right out of the gate up 240%, and midday it was up 165%. My question to you is this indicative of the deals that are getting done in the market right now? Has there been a change? And is this representative of what you think strategic deals could look like for Q as you look at the indications that we talked about before?
Sure. Let me address the question from a market perspective, and I'd like Anish from a technical scientific perspective, because the field evolves and we go through different periods. It's not surprising to those listening on the call, the biotech sector has gone through a contraction over the past couple of years. And it's because it was overstimulated. There's too much capital went in an undisciplined manner, including pharma. They were investing in these IL-2 plays, and the failures kind of jolts the system. And now you're seeing that the dust has settled. They're looking for where there are rational approaches going forward. This transaction you just highlighted, it's indicative of an appetite pharma. They have a lot of cash. They need to have therapeutic modalities that give them an opportunity to address these unmet medical needs. We're in discussions now with multiple parties. We're not just reacting to a situation. We're trying to play this out strategically and thoughtfully. We want to have strategics that align with our vision, understand the platform, are going to deploy the requisite resources in a very deliberate manner. We don't want to license the platform. That's the beauty of the platform. We can license a program and still retain the significant upside of the platform.
Right.
Where we do license, for instance, in autoimmune with Ono, we structured that transaction where they're subsidizing the preclinical development. We can opt in for co-development in the United States, and they continue to pay milestones, which
So
in
you
essence
preserve
covers our
that
cost.
upside, and that's again, if I'm hearing you right, that's the strategic pursuit. That's your strategy for business development: is to preserve the upside, not just license out, but to find ways that you participate in the upside, and prevent dilution down at these levels.
Yeah.
Essentially, I mean, would that be accurate?
That's accurate, and I want to emphasize we did not evolve the company over this past 7 years to just license out programs where we're a preclinical discovery engine. We have a very capable, translational focused clinical group.
Yeah.
Headed up by Matteo Levisetti, our CMO, and he's built this really solid group that knows how to design these trials in a very effective way. And the quality of our communications, I think, with the FDA underscores that.
Yeah.
So we want to continue evolving that group.
And if I'm correct, Matteo was at Mirati before here, right? Mirati got taken out for about $5 billion for a single target, really, on KRAS. And Cue has developed 4 potential targets around KRAS. Is that accurate?
Yeah, so Mirati's, so that's right. Matteo was at Mirati. In fact, he was foundational to setting up their immuno-oncology programs. Before that, he was the head of development for immunology for Roche in Basel, Switzerland.
Yeah.
Before that, Merck, Pfizer, so has had a lot of experience.
He might be the smartest guy.
He might be. He most definitely is the smartest guy. But I think what's remarkable is that's a small molecule for a mutation for G12C. It's a mutation to a cysteine.
Okay.
KRAS has dominant mutations are some other amino acids to which we have made immunostats, and there's 4 different flavors of that. And that, as Dan mentioned, has caught the attention, their preclinical assets, but of parties that are genuinely interested. And remember, KRAS is the most highly mutated proto-oncogene in pancreatic cancer. 80% of pancreatic is KRAS mutated. About 40% of colorectal is KRAS mutated.
So very big end market.
Good chunk of lung, and a good chunk of lung cancer, which is where the Mirati molecule is approved, as you're well aware. So you look at the indications for KRAS, Lou, between lung, colorectal, and pancreatic. I mean, that's a massive opportunity.
Massive opportunity. So let's just time constraints. Let me transition before we go to Q&A and ask this question. What keeps you guys up? The data is maturing as the science would suggest it would have. It's playing out in multiple now assets, 101 to 102. So what worries you? What is the concern as you wake up and come in an increasingly cold winter climate in Boston?
I'll go first. With my concerns, and I'd say it's my frustration, is that we have demonstrated a platform that clearly transforms the way cancer can be treated. If you think about it, if you're a cancer patient, you've been diagnosed, the idea that I can prime my immune system to attack cancer and it has memory, so that even when I'm no longer receiving drug in the future, I still have activated T cells in my body.
I think that's an important point that I don't think many people understand, because if we go to the monotherapy study, if you look at that bar graph that you used to show, it showed time on drug and then how long they've survived. There was instances of maybe 2, 3 doses, 4 doses, and they're extending out to plus a year.
Yeah. The patient's own immune system is the therapeutic. Our drug enables it, and that's an important qualifier. So my frustration is it's basically capital constraints with the capital market, where our stock is right now, because the whole market got depressed. I don't think there's a real understanding of how profound this data is yet. And I think pharma is starting to give us that attention. And that's where we're looking to pharma for capitalizing. We're not looking at diluting shareholders in a future raise, but we need to enhance our bandwidth so that we can develop this platform and treat multiple cancers. These therapies are basically waiting to be developed.
Yeah.
So
this is more now a business development exercise than science. I mean, obviously the science has to be concurrent and moving down.
Foundation,
right? Science is a foundation, but now you've gotten it to a point where you can monetize that science in pieces to increase shareholder value, reduce dilution, which I think is what everyone that's an investor, and you both, I might add for everyone's benefit, these two gentlemen own meaningful positions. They're not here just for the fun of it, right? So you want to see the success of this, just like all of us. We're aligned to win in the same way.
Yeah, and look, when you think about this as a therapeutic modality and a platform, time is not the friend of patience, and it's no one's friend. We need to be moving, and it's momentum. So that's my frustration.
Yeah,
I would like to be able to hit this in a much broader swath.
I'm glad you didn't say your frustration was Anish. We'd have to have different
That does
conversations.
happen.
So Anish, from a science perspective, what are the things that you think are limiting or challenging, and how do you overcome them?
Yeah, not any major frustrations with the science, Lou, because it's played out. I think for me, the most important element as the head of R&D is to continue to maintain focus and discipline. We've got to just continue to execute and generate the data. We've never dabbled. We could have done 20 different things. We've stayed true to 2 molecules in the clinic, 5 indications, and a rich preclinical pipeline. We haven't done IL-2, IL-18, IL-12. We've just been very disciplined. I'd rather do 3 things really well and really deep than dabble and try to boil the ocean, and that gets you nowhere. So we've maintained that. The second thing is we've always been very diligent. We've sort of not gone into this rut of being intellectually lazy. We want to make sure we're constantly not paranoid, but on the front edge of this, leading this. I think there's a change that's already emerging in the space in terms of immunotherapies. And I think it's a great position to be in for us. And you're seeing with the data now with about 100 patients dosed. And let me make the point that this is a first-in-class platform in man. This story could have been over very quickly by your second dose group if this molecule was not well designed.
Yeah, I think in the last conference call you said first in class, and then first best in class.
Best in class. There's nothing else. I mean, now people are doing this. Now people are trying to replicate what we're doing using T-cell receptor specificities to dial in. We have been ahead of that curve.
Yeah.
But we should remind ourselves that this platform and technology has never been in man. There's not a historical precedent to build on as antibodies have had,
Right.
And then you're just
you're forging new ground, creating a paradigm shift.
Yeah.
All right. Well, this has been extremely instructive. We're going to open up the questions and answers right now, briefly, maybe 2 or 3. So if anyone has any questions, we're going to take a pause for about maybe 30, 60 seconds. You can email them to community@publicventures.com. Again, community@publicventures.com, and then just give me a moment to start collecting some of those that might be worth responding to here in this forum. So again, the address is community@publicventures.com. We got one here that gets to the specificity. So you target, in the case of Q101, it's HPV positive HLA-O2. HLA-O2 is a specific allele that covers, I believe, 40 to 50% of people in the world.
That's right.
Do you investors, and I've heard this before too from several investors, they think the fact that you got to tailor it for each different allele means it's a lot longer time to develop, a longer time to get to market and realize the value. Do you see that as a limiting factor? And then this question here is, do strategics or partners see that as a limiting factor?
So one of the beauties of our platform and the strength is the modularity loop. So we have already, for example, if we wanted to make analogs of Q101 on other HLA alleles and capture the rest additional 40% of patients, we could do that. That's just a matter of resources.
Yeah.
It's not a matter of if, it's a matter of when.
Right.
And if you think about it from the perspective, if you had resources, you could do this all in parallel. The reason AO2 makes sense is because it's the largest chunk of the patient population the markets we're interested in. North America, Western Europe, getting 50% of that is pretty significant.
Yeah. So the allele is really just a geographic.
It's a geographic
right? So it's not anything specific to the molecule itself.
No, it's not. So for example, if we were to develop an analog of 102 or 101 in Asia, in China, Japan, we would do A24. China's 50% of A24.
Yeah.
That's the AO2 equivalent in China.
Gotcha.
And in fact, we've already made these molecules. So, that's the modularity. This is not a limiting. This is actually exactly the right way to expand personalized medicine, is you do it tailored, but you do it tailored after these dominant alleles.
Not like CAR T, which is one-to-one, where it's the
One-to-one.
patient's
Own cells going back
Yeah.
into your own cells. So now the other important aspect of that, and I'm glad you asked this question, is between 4 to 5 dominant alleles that we know in any geography. If you made to a tumor antigen 4 to 5 alleles, you can capture up to 80 to almost 90% of
Okay.
patients.
So not really limiting them.
Not really limiting at all.
All right, a couple more questions will come in, and you addressed this on the last conference call, but I think it's worth repeating here. What are some pivotal events, catalysts that you see in the year ahead in 2024?
Sure. First, we have a meeting with the FDA in January. That's important because it establishes that, again, foundational data that has implication for the combo study, et cetera. And it actually is an education between the two parties, establishing a common understanding of the platform, just like we had with 101, lending itself to de-risking 102. Further data readout on combination that's continuing to mature, looks even more robust. And then with 102, we should be able to report, let's say by mid-year into Q3, the results of what we're now observing in the early dose escalation. We're going to start the patient expansion in the first half of 2024. That is really important data because we have 4 indications we're surveying, and we're already seeing anti-tumor activity emerge, even in the dose escalation. And importantly, we're in multiple strategic discussions with potential partners. And we're expecting that in the first half of the year to clarify what route we want to go down.
Okay. So no, that's good. I was fumbling here to try and bring this slide up. Yeah, so.
I think I remembered most of it.
That's a good thing.
Q401, actually, also by mid-year, we have a potential of selecting a clinical candidate, and we can opt in for co-development.
And 401's an autoimmune.
That's an autoimmune.
Unspecified autoimmune disease right now, or is that something that's been disclosed?
We are in discussions to actually identify the indications where we would first go in with obviously owners, our partner here. But in contrast, the one thing I should mention for 401, Lou, is 401 essentially has a massive potential in a wide range of autoimmune disease. We're talking about the market opportunity. Rheumatoid arthritis, inflammatory bowel disease, psoriasis. So these are pretty significant marketplaces.
Yeah. Again, this is dovetailing into market opportunities, strategic discussions, catalysts. CAC runway is about $54 million at the end of last quarter was the cash balance. Burning historically somewhere 40 to 45 million a year. Is that still accurate? And
that's still accurate. Let's say around 45 with the clinical activities increasing a bit. So we're confident we can get through 2024 and into 2025.
Okay.
With no additional capital infusion. The objective is we are not looking at raising capital with a stock offering, but we're looking at doing that with a strategic partnership.
Okay. So if we, again, and I'm not making any assumptions, but just using current market deal of today, a 60 million plus a 30 million near-term milestone, that buys you, if you had a deal like that, that's 2 years of additional.
That's a rational assumption.
Okay. I think got a few more here coming in. Let me just read real quick. There's a question regarding the business development. I'll paraphrase here. Is just the discussions that are going on. Any insights into pushbacks from partners that they only want certain assets versus other indications, or where they want more than one? And how do you look at that strategically and saying, hey, here's what we're willing to offer. Here's what doesn't make sense to bundle, and vice versa.
It covers a spectrum of discussions. We have pharma companies that are specifically interested in, for instance, 101, companies that are interested in 102, companies that are interested in both of them. And we have dialogue on platform, and we have dialogue on autoimmune. So we have the spectrum, and what we're trying to do is assess what represents the most attractive opportunity for giving us access to resources, retaining upside, and what does the support level look like.
Okay. All right. Well, I think we've got some other questions that people want to nerd out and get really into the science, and I can't do that. So we'll make sure we connect them with you after the fact. I know that you have done a tremendous job of putting some of those resources on the website as well. If people want to dig into the science more, some of the published,
Yeah, we've all our papers are on the website, and we've made it a cadence, Lou, that whenever we present it at a scientific meeting, those presentations are always posted on the website.
And the next meeting big is JP Morgan in a couple of weeks. And then the next
Yeah.
scientific meeting would be what, April-ish?
Well, we have ASCO
Yeah.
mid-year.
Okay.
The cadence we've had is we've done a mid-year at ASCO and then year-end at SIPC. That gives us a good opportunity to pull those.
All right. Last and final question that has absolutely nothing to do with this, because I like to end this up, and I'll lead to make it easy. I always go to new cities that want recommendations of where to eat. So I'm Italian, pizza, there's no better food, and we're not in Connecticut, so it's not a pizza. So pizza in New Jersey is the Reservoir in Boonton. Best pizza, hands down, don't miss out. What's your best recommendation? Food can be anything, doesn't have to be Italian. You could, go against our heritage, but
Italian is a place in Boston called Toscano's, which is outstanding.
all right.
And pizza, it's Regina's.
All right.
Anish?
Anish?
Toscano's is outstanding, Dan. I've been there with Dan. And I'd say for steak, Moo's excellent.
All right, there we go. There you go. You got your stock information, science information, and some restaurant tips too. I want to close out today by thanking both of you gentlemen. Really appreciate you making the time right ahead of the end of the year. I know there's a lot of busyness, and I want to thank all of you who had the opportunity to join us today. This is our inaugural conversation. We're going to look to do this moving forward at least once a month. So I thank you for your continued loyalty and support. And if you have any additional questions or things that constructive criticism, we'd rather know what we do bad and we can improve than just give us attaboys. So email us at community@publicventures.com. And we look forward to speaking to you in the near future. Thanks again.
Thank you.
Thank you, and want to thank everyone for listening, and your support allows us to do this important work. And happy holidays, and everyone stay safe.
Very well. Yeah. Happy holidays.
Take care, everyone.